Thank you so much for this exciting panel. And now we're coming to, in fact, the people that will be hopefully using our models by making their drugs available. And this is in fact the last panel of the day, which is the company panel. So I'm very excited to have a few representatives from pharmaceutical companies with us this afternoon. Not every company that is working on NF2 was available for this panel, but we have a few good friends. So with further ado, what I would like to do is maybe just go around the screen and ask every company representative to introduce themselves, maybe talk a little bit, if possible, what kind of NF2 target you're working on, whether you are in the preclinical or clinical phase. Just give us talk a little bit about yourself. So I just go around the screen. Mark, shall we start with you from Alira Biotech?
Yes, thank you, Annette. Can you hear me?
Yeah, perfect.
Perfect, yes. Yeah, so my name is Mark Benedict. I'm CEO of Alira Therapeutics, and we're actually focused on the other end upstream of Merlin. So we're focused on inhibition of GTPases that stimulate PAC1 specifically and very precisely to complement and use those inhibitors to complement the Merlin mutation, which drives NF2. We're in the preclinical stage, and we acknowledge CTF's wonderful award with which we're collaborating with Wade's group at Indiana.
Neil?
Hi, hope you can hear me. This is Neil Kirby. I'm CEO of Mulberry Biotherapeutics. So we're leveraging the work that was started by Gary Brenner at Mass General in looking at a bacterial therapy approach to NF2. So we are using an attenuated strain of Salmonella typhimurium, and we intend to be in the clinic in early 2023. We have one of the advantages here is that the Salmonella strain that we're using as our lead candidate has actually been in man before for other indications in melanoma and so on. So that's the starting point for our clinical study, which I say should be initiated in early 2023.
Thank you. Glenn.
Thanks, Annette. My name is Len Post. I am CSO of Vivace Therapeutics. Vivace was founded by Qinlang Guan and Sheng Ding to specifically discover drugs that work on the Hippo pathway, which, as you probably know, controls the transcription factor YAP, and in the middle of the pathway sits NF2. The compounds that we discovered through a high throughput screen turn out to inhibit the polymethylation of TEAD, which is the partner of YAP. And we have optimized that compound and have our first compound in phase 1 right now. We're in a dose escalation phase. It's an oncology phase 1 in the US and Australian sites. Typical oncology phase 1, taking a broad range of solid tumors, although we're encouraging our investigators to look for tumors that have NF2 deficiency. And once we get our dose worked out, the plan will be to study specifically NF2 mutant tumors.
Thanks, Glenn. Glenn?
Hi, good afternoon, everybody. So my name is Glenn Morrison. I am a VP of Clinical Development at Recursion. Recursion is a clinical stage biotech company that is really focused on industrializing drug discovery, really to drive the efficiency of bringing effective medicines to patients who need them. It is a disease-agnostic approach, but our lead compound for NF2, REC-2282, is an HDAC inhibitor, small molecule. It is CNS penetrant, orally bioavailable, and we are clinical stage. As I mentioned, we expect to initiate a phase 2/3 study in patients with progressive NF2 meningioma, hopefully this quarter, and look forward to advancing this medicine.
Thank you. Thank you so much. So one of the things that we have discovered in the NF2 community is that although we have, on the one hand, we have now that wonderful platform trial that is running in the US called the Intuit trial that is run in 6 sites, led by Scott Plotkin. We're developing a similar platform trial in Europe for both NF1 and NF2 through a funding from the Innovative Medicine Initiative. It is hard to get companies, or let's say to convince companies that NF2 is a good indication, right? They say, even if we say we have preclinical data to see that this drug is really working and we really would like to be able to offer this to our patients, we regularly get, unfortunately, a nah. So what would be interesting, I think, for our community to understand is what brought you to NF2 and what is kind of, for you, why are we attractive to you as an indication? And hopefully we can use that as an incentive to start attracting other companies. So I don't know who wants to jump in first. I don't want to start picking people, but I think it would be interesting for our community to understand that. What makes pharma decide to enter into our space, which of course we're very thankful for.
I'm happy to start. I think that if you're working in a science-based organization, you have to let the target pathophysiology drive where you're going to go. And like many other rare disease indications, which have kind of commonalities in biochemistry with oncology indications, I think NF2 presents an opportunity to be in a place where there are regulatory incentives and other incentives in Europe and the US, which can benefit a company overall in a big way. And so I think just from a scaling standpoint, if you're selling products for hypertension and pain management, et cetera, and those franchises are very, very large, you've got resources that are going to be committed to equivalently large potential franchises. And just from a financial quantitative standpoint, you're going to have a cutoff at some point for a hurdle rate that makes sense. And that's probably why I think big pharma is doing what it's doing. They basically use small biotechs as off-balance sheet research vehicles. And when the products are de-risked, if they show promise, they'll buy them, either by buying the company or doing a licensing deal.
Anyone else who wants to comment on this?
Yeah, I'll just make a couple comments on that. For Vivace, it was also just very science-driven process. When we found out that our compounds worked in NF2 mutant cancer cells, it sort of just pointed in the direction of what else could we use our compounds for. And we had the good fortune that actually one of our investors encouraged us to talk to CTF. And that really made all the difference in the world for us, because we didn't really know where to go to start to study these. NF2 models aren't available at CROs, which we're dependent on. And, CTF really made the right introductions for us so that we could start to get our compounds tested. And David Parkinson mentioned that he's generated some very nice data with our compounds. So it was a matter of following the science and finding a path that the models were accessible. And that's not easy without some help in a process that CTF provided for us.
Yeah, and for Recursion, every program at Recursion is a product of the Recursion operating system, really this tech-enabled drug discovery platform. And our programs target diseases spanning several therapeutic areas, but they have two common criteria. One is that the disease-causing biology is well-defined, but maybe the downstream effects are poorly understood or maybe undruggable. And second is that there's a high unmet medical need, no approved therapies, and significant limitations to existing treatments. This is really where rare disease, I think, fits nicely. For us, following this approach, we identified REC-222 as our clinical candidate, and in preclinical models, it was shown to restore deficits in some models of NF2. Fortunately for us, there's also some clinical data for this compound suggesting that REC-222 may have benefit in patients with NF2. And so really that's how this program, I think, was born at Recursion.
Neil, do you want?
Yeah, sure. So maybe Mulberry is a little bit different than all of the other companies that have presented so far. I think that, we were formed around an asset that seemed to have promise in NF2. It wasn't an oncology program that was then moved into NF2. And actually, it may be going the other way in that we're now generating data in other tumor types, NF1 and meningioma. So we are focused on NF2, and that's will be the definitive step for the company is proof of concept in NF2. And hopefully we can use that and leverage that data in other indications as well.
Thank you. So to turn to kind of flip the question upside down now, what would you think would companies refrain from entering into the NF2 space? What do you see as the biggest, what have you seen as potential obstacles, or what could you think your company friends would see as potential obstacles to enter into our space? And then of course, my immediate next question after that is, what can we do to become sexy?
Maybe I'll start here.
You
Sure.
want to jump in?
Yeah, I mean, I think for me, one of the main obstacles really is the nature of the disease itself. I mean, these are slow-growing tumors, and so any treatments that we think of obviously have to be taken for a very long time, maybe for the duration of the patient's lifetime. And this means that clinical trials that we're doing that are really focused on regulatory endpoints can take a very long time. As well, the therapeutic window that we typically associate with oncology drugs likely won't apply to the NF2 patient population. Lots of oncology trials fail, mainly because of efficacy, but a large portion of those still fail because of safety. And I think this makes getting medicines to patients a little bit more challenging in that we're trying to find compounds that are effective, but at the same time safe for chronic use. And I think that's one of the main obstacles for companies really getting into the space. Studies take a long time and a large investment to be able to demonstrate efficacy and a reasonable safety profile, especially in this type of indication.
So you're putting yourself a little bit in the hot seat now, right? So I know that during the prep for this panel, we were discussing and saying that, yes, I still remember from my pharma time, my vice president would always say, you want to kill a drug as quickly as possible. So you don't want to have to wait a year for a preclinical trial to be able to decide. So now I'm going to throw out the real hot stone here is, could we do without animal models? What is the data that would need to be provided to convince the regulators that we may not need a 1-year lasting animal study, and that a combination of some animal work, obviously, but how much can we do and how much could we do in a faster way with you guys who have experience now in the NF2 space? Let me put it a little calmer.
So perhaps I could just jump in there. I think that the whole question of animal models in rare diseases it's a difficult issue. And of course, the most important model is man. And the idea to get to trials in man as quickly and as safely as possible, I think is the way to go. You've got different audiences, of course. You have to persuade the FDA that you have sufficient efficacy in a model that gives you ideas that where you can take it into the clinic. But then you also, as a company, you need to go out and raise money. So VCs have a very different outlook on animal models, and it's often VCs and investors who want to see additional animal models, whereas the FDA may be happy with one model rather than 2 or 3. And so, this whole notion of translation from animal models to humans is a tough one. And certainly I've been in rare disease drug development for over 30 years, where very often for certain diseases, there aren't any animal models at all. Does development go faster in that case where there are no models? It'd be an interesting analysis to do, I guess.
Just as an aside, Neil, I've spent many years in Alzheimer's research, where we've cured Alzheimer's in mice maybe 1,000 times with animal models that don't translate well to the human condition. So there you go.
Yeah,
Yeah, if we were developing drugs for mice, we'd be done by now, right?
I would echo Glenn's comment, and I also have been working in Alzheimer's, in R&D and BD for quite a long time. It's a good parallel in that, the course of disease is so long that your ability to generate data that shows efficacy is just hampered by the costs and the timeline, and so the FDA doesn't really have a separate track for those types of diseases. And what's happened over the years is that any disease that has a very slow course of development that requires, say, daily dosing with a very wide therapeutic index are now the diseases for which we don't really have many cures or treatments. So that's partly a casualty of the system. I think, at the end of the day, we're all hoping that somehow we impact Merlin and that whole pathway, et cetera, from some direction or another. With what we're doing, but as Glenn pointed out, I literally remember going to the board of directors at Elan Pharmaceuticals, and I was told, we've cured Alzheimer's. We don't need to do anything else. And I said, please, let's do this deal because it's an IND stage product. But I was dismissed. So it's still very much a black box.
Yeah. So in fact,
sorry.
No, the idea that I wanted to mention is to say, should you do at least tox studies in animal models, obviously, but then if you know that the drug actually reaches the target, so kind of a PD tox study in an animal model, and then have your biology in your spheroids or organoids or something that can give you a quicker decision, right? Kind of a combination. I don't know. I'm just.
Are you saying that you would try to prove safety and PK/PD in a sense, and outside of an animal model, and then go to the animal model for some additional efficacy work, or how would you?
No, I would do, but this would be me, right? I would do the PD and tox in the models, but then in fact you're making sure that your drug actually reaches the targets, and then do your tumor biology potentially outside the animal models. But I don't know. I'm trying to accelerate the process, right, and make us more attractive to pharma people. But sorry, Len.
No, I mean, I was going to say actually something very similar, Annette. I think that I think that a key to it is to break it down into steps. It's a big leap to go all the way from an in vitro model to a multi-year clinical program. But there are pharmacodynamic endpoints in relevant animal models that some of the people on this call have characterized. So maybe you wouldn't need the long, big clinical endpoint study in the animals, but pharmacodynamics showing that the drug is doing what it's supposed to do in a relevant model. And I think that also might go for people. And I know this is much harder. If the community could come up with a biomarker so that there was some way to get a signal that the drug was doing something in people, short of a multi-year clinical program, that could also lower the activation energy for a company willing to take a compound into an NF2 trial, I think.
Yeah, Len, I completely agree. I think the whole biomarker story is one that needs to be explored in more detail. Of course, there's still the issue with the FDA of Subpart H approvals and, likely to be predictive of clinical benefit. And, my definition is quite different than theirs probably, but biomarkers are going to be important.
Even if it's not an approval endpoint. Back to the point that was made earlier about how companies need to justify to their investors and spend the money, that biomarker showing that there is activity, even if the FDA is going to require the clinical endpoint, or that'll be, of course, be a negotiation, but it could be a really important step in the development path.
I think along those lines, one of the opportunities perhaps is really to work to elevate some of these non-imaging endpoints to approval status. Are there patient-centric endpoints that could ultimately support approval and get medicines to patients faster? Along the lines of the accelerated approvals that we've seen, where perhaps you get an approval based on a PRO or some other patient-centric endpoint with additional studies that are required and able to demonstrate that patient. At least you can get the drugs approved. Patients can start using them, and we can conduct these longer term studies that might be needed from that full approval perspective.
Yeah, and I would add to that, again, going back to Alzheimer's disease, looking at, what is your label going to say? And Glenn, I'm going to shamelessly crib from what you said the other day. But basically, when you look at Alzheimer's, are you delaying progression? Is that a label claim? Are you, enhancing activities of daily living on top of some other measurable endpoint, which is still very significant for patients? So I think that, at the end of the day, looking at it from the clinical side and the patient side is very important. If you can, maintain, defer hearing loss or reduce hearing loss, maintain other kind of critical functions in a patient, that should still be something viable that you should go forward with. And if there are no toxicities or any other issues, I'd like to see drugs like that go forward in NF2.
Absolutely.
So Mark, you brought up in the prep call as well, the value of data, data collection, real world evidence. Do you want to talk a little bit about that, about the value of helping that in getting more attraction to the companies for NF2?
Yeah, I sit on the board of Rare Science, which a friend of mine started several years ago. And I know she has affiliations with Recursion, Chris Waters. And it's an interesting kind of focus at that entity because the focus is really to aggregate genomic data, clinical charts, et cetera, and really kind of pull together any piece of information you can that's quality about patients, and work with patient advocacy groups also to kind of mobilize that data, create a federated data source so it can be used by pharma companies more easily. And so, I think those efforts are almost more valuable in terms of bringing clinical attention to the disease than early stage discoveries about different biochemistries, et cetera, because they just kind of on the other side of the equation, they help facilitate kind of moving things through the clinic in a big way.
So there was a question in the chat. There was a question for Len. Have you tested TEAD inhibitors in Schwannoma models?
At Vivace, we have not, but David Parkinson has. And in fact, he presented, Liam Larava from his lab presented at the CTF conference last year, some of the first data in the mouse model showing that there was at least pharmacodynamic activity of the TEAD inhibitor in the Schwannoma mouse and the periostin Cree that was discussed earlier.
Okay, thank you so much. So I had one more question, in fact, to all of you, and maybe just, if you can just spend maybe a minute of your time to talk about that, is that what would you advise a community like us or a foundation like ours? And this is kind of nicely going into our what could money do, right? Our next discussion. What could money do? So what would you think a foundation like ours, like a research foundation, what can we do? Should we raise more awareness about the biological targets that could potentially be important for NF2? What would you invest in?
So I see the opportunities on two fronts. One I mentioned already, which I think patient advocacy can play a really important role in defining what's meaningful for patients and really talking to FDA and other agencies about potential other endpoints, along the lines of what Mark was saying. Are there medications that can be potentially they're going to perhaps result in tumor shrinkage down the road, but a much more proximal outcome is going to be on some sort of clinical benefit, hearing loss, et cetera. So I think that patient advocacy can certainly play a very important role in having those conversations with agencies. I think the other opportunity is, I think it was talked about a little bit earlier, but some of the preclinical models that are available, really continuing to develop and validate these models so that there's wide access for companies to be able to leverage them in their drug development practices.
Yeah, I think that's key. I was going to say to really define what both the preclinical and the clinical pathway would be, to develop a compound and then make those tools available to the community in a way that companies of all sizes have access to them.
Yeah, I would add to that, and I would also say, just from a FDA standpoint, the past couple of years, the FDA has kind of changed their tune a little bit. Where now they're at the point, certainly with Vertex, as we mentioned in our prep call, they basically allowed Vertex to expand the label for cystic fibrosis patients based on in vitro data. So I think given that precedent, which is very far on the other side of what we're talking about, that kind of model should be kind of pressed to the FDA's attention and kind of be used as a driver to see where other companies can go to get products approved without the onerous lengthy trials that we're talking about, and think about. I would also echo what Len and Glenn said, which is kind of open sourcing the toolbox for preclinical use and really trying to make sure anything that's being offered up by CTF has immediate clinical relevance to expediting clinical studies.
Yeah, I'd like to add to the statement about patient advocacy with the FDA. I think that it is really important because sometimes the agency they can't be aware of all of these rare diseases. And, I was involved with the epidermolysis bullosa, and, they went and spoke to the foundation, went and spoke to the FDA, and there was a guidance document came out very shortly thereafter. It wasn't very helpful, but it was a good starting point. And there was an increased awareness in those patients. I think the other piece that's important for companies to develop drugs is access to patients. To know that there are sufficient patients to be able to do a study quickly and safely is important to most rare disease companies as well.
Yeah. So that I think that nut we are cracking through both Intuit and now in Europe as well, we're building that similar platform, let's say enterprise. There is one question, and then I think we should close the panel, from Adam Goodkind. Can you speak towards machine learning powered drug discovery, specifically its potential for rare disease and more specifically Schwannoma? So that, I feel that's probably a question for Glenn, right?
Sure, I can speak to it very, very high level. Recursion is a tech-enabled biotech company, of course, and we do leverage machine learning and AI algorithms to identify clinical drug candidates through what we call Phenomic Screening. And so what this means in very high level terms is that our team of scientists conduct a number of experiments where they can knock out genes in human cells and generate a phenotypic map based on imaging, and then use that information about the genes, molecular pathways, et cetera, to identify matches, either those that are similar in phenotype or those that can reverse the phenotype. And in this way, we can identify biologically interesting combinations and targets that may identify effective therapeutics. And so this is, I think, can be really powerful in the rare disease space because we can look at monogenic aberrations and look for either novel compounds or compounds that may already exist that can reverse those phenotypes and might be potential candidates. And then we work to validate those targets and then hopefully move them into the clinical study space.
Thank you. Thank you so much. Well, thank you everybody on the panel for this honest and open discussion. I was very excited by the fact that you guys accepted, and I hope you can go and convince other companies to join this wonderful community. And I really hope that you will be present at our NF conference in June, because we need these ambassadors to kind of talk well about NF2 so that we get more treatments to our patients.